We will cover both chapters 6 and 7 this week. While both chapters explain cell division, chapter 6 discusses mitosis and chapter 7 discusses meiosis. Mitosis is a type of cell division that happens in single-celled organisms as a means of asexual reproduction, and it happens in sexually reproducing multicellular organisms such as plants and animals, and is used for growth, maintenance, and repair of cells and tissues. Notice a multicellular sea urchin dividing from a single cell to 16 cells, then ultimately developing into a full-grown adult. Although the sea urchin is a sexually reproducing organism, it uses mitosis to divide cells for growth and maintenance. Before we dive into cell division, it is important to understand that cells carry genetic material. All of an organism's genetic material is known as a genome. Notice in the left diagram, in prokaryotic cells, which were cells lacking a nucleus, DNA is found in a region known as the nucleoid, and the DNA is circular. Prokaryotes can also have small additional loops of DNA known as plastids. In the right image, eukaryotic cells, which were cells that have a nucleus, DNA is linear or in a line and found in that nucleus that those cells have. Deoxyribonucleic acid, or better known as DNA, is organized in the eukaryotic cell in matching pairs called chromosomes. A set of chromosomes is referred to as homologous. In homologous pairs, one is inherited from the mom and one from the dad. The blue and red chromosomes would be a matching pair. Chromosomes are wrapped up in coils of DNA strands. On each strand are segments known as genes. It is genes that determine the characteristics of an organism. This could be eye color, fur or no fur, or how tall an organism grows. The specific location of a gene on the strand of DNA is known as the locus or loci for plural. Loci are the same for specific genes in the same species. For example, the genes that code for eye color in humans would be found on the same chromosome and in the same region on the chromosome on both parents' chromosomes. At the end of this chapter, we will discuss cell division in prokaryotes. Watch the linked video for an animated representation of DNA. Chromosomes are represented by the letter N. In somatic cells, all cells except for sperm and egg cells, cells have a pair of chromosomes, one from each parent. This set of chromosomes is sometimes known as the diploid number and is represented using 2N. In sex cells, also called gametes, which refer only to egg and sperm cells, the cell only carries one of the two chromosomes. These cells are referred to as haploid as they only have one chromosome of the pair. Notice in the karyotype image of the chromosomes from a human cell, there are 23 sets. 23 chromosomes from the mom and 23 from the dad. This equates to a diploid number of 2 times 23 equals 46, and a haploid number of 1 times 23 equals 23. Watch the linked video comparing diploid and haploid cells. When cells divide, they go through the cell cycle, which consists of two main phases, interphase and the M phase, M for mitosis. During interphase, the cell grows larger and duplicates DNA, while during the M phase, the cell splits into two daughter cells. Before going through all the steps of the cell cycle, watch the linked video to help visualize each step more clearly as we proceed. Most of a cell's life is spent in interphase, the time when no cell division is occurring, but when regular metabolic activity occurs. Interphase is divided into 3 subphases, G1, S, and G2. The G stands for gap, which is when no DNA is being synthesized. The S phase is when DNA is being synthesized or replicated for cell division during mitosis. Chromosomes in this phase are copied into two sister chromatids. Finally, G2 is a phase for protein synthesis, which helps prepare the cell for mitotic division. Watch the linked video to help better visualize the difference between homologous chromosomes and sister chromatids. After interphase, a cell will enter mitosis, which is divided into 4 stages, remembered more easily with the PMAT abbreviation, standing for prophase, metaphase, anaphase, and telophase. At the end of mitosis is cytokinesis, which is the final division step forming 2 daughter cells. Cytokinesis means cell movement. As a visual representation of the entire mitotic process, start at the far left with prophase and notice how the changes in the red chromosome with each step ending with cytokinesis, where one cell becomes 2 daughter cells. Now we will look at each step individually. During interphase, before mitosis, chromosomes are loosely arranged, similar to a bowl of spaghetti noodles as in this top image. When prophase begins, the chromosomes condense and become more visible, looking like an X. The membrane surrounding the nucleus begins to break down. The centrioles, which help pull the chromosomes apart, move to the poles of the cell, and the chromosomes are connected to the kinetochore, which is a set of strands connected to the centrioles at the poles. During metaphase, the chromosomes align down the middle. This is easy to remember because both metaphase and middle begin with an M. Each sister chromatid is attached to a fiber which is connected at opposite poles. During anaphase, the 2 sister chromatids are pulled apart from each other to the opposite poles. This set is easy to remember because anaphase and apart both start with the letter A. The cell begins to elongate, preparing to split into 2 daughter cells. During telophase, the chromosomes are now at the 2 opposite poles and are each surrounded by a new nuclear membrane. At this point, there is still only 1 cell, but it has 2 nuclei. You can use the T in telophase and the T in 2 to help remember this. The final step of mitosis is the step of cytokinesis, where 1 cell has divided fully into 2 daughter cells. In animals, there is a cleavage furrow that splits the cells, while in plant cells, a cell plate must be formed in order to duplicate the cell wall in plant cells. Now that you have walked through all the steps of mitosis and cytokinesis, watch the cell cycle video again to better visualize the process. Sometimes somatic cells do not need to divide and will therefore halt in phase G1 and are said to be in a G0 phase. They will stay in that phase until an external trigger signals the cell that it needs to divide. Some cells, however, reach a maximum size and do not divide any further, such as heart cells. Any cell that divides is the ancestor to potentially trillions of daughter cells. If a mistake occurs during division, those mistakes, known as mutations, could be passed down to all future cells. In order to prevent errors from being passed on, the cell division process has several checkpoints. The checkpoint near the end of G1 checks to make sure there is no damage to the DNA and that the cell is of adequate size to replicate. At the end of G2, the checkpoint is designed to assess proper chromosomal duplication as well as ensure no damage has happened to the newly duplicated DNA. And finally, the M checkpoint is designed to ensure that the sister chromatids will separate by confirming the kinetochores are properly attached to each pole of the cell. Sometimes the cell's checkpoints fail and do not catch errors. When that happens, a mutated cell can divide uncontrollably, causing a tumor. Cells have two regulator genes to help prevent tumors, but they can also fail. Proto-oncogenes help detect cancerous cells and arrest division. If, however, they fail, the gene turns into an oncogene, promoting cell division and thereby promoting the growth of a tumor. Another regulator is a tumor suppressor gene, which are designed to suppress the growth of tumors. But if they fail, they will become inactive, allowing unregulated growth of a tumor. Watch the linked video for an animation of the two types of genes. So far, we have discussed the replication of eukaryotic cells. Eukaryotes use mitosis for growth, maintenance, and repair within an organism, but some organisms use mitosis for a method of population reproduction. Prokaryotic cells replicate by asexual means such as binary fission, which produces an exact copy of the parent cell and can result in many copies in a short period of time. When single-celled organisms reproduce, the process begins at a point known as the origin of replication, where DNA is duplicated, moving away from that point. Notice in step 1 of the diagram. The cell begins to elongate in step 2. Then we'll begin the splitting process with the help of a cleavage furrow, step 3. And once the two DNA loops are on either side of the furrow, the cell forms a septum, which separates the two cells, resulting in two daughter cells, as in steps 4 and 5. To see an animation of this process, watch the linked video. Alright, so that's it for Chapter 6. However, Chapter 6 covered somatic cell and prokaryotic cell division, but this week we're also going to talk about what's in Chapter 7, which is cell division of gametes, or our sex cells, the sperm and the egg cells. So make sure that you've read Chapter 6 and Chapter 7, and definitely go back and review the end of chapter questions.